Welcome to the world of computational biology, an interdisciplinary field that beautifully merges computer science and molecular biology. Here, we explore the fascinating applications of computational techniques in DNA sequence analysis and bioinformatics, unlocking the secrets hidden within our genetic code. So, what exactly is computational biology? It is an interdisciplinary field that uses mathematical models, computational simulations, and data analysis techniques to study biological systems and relationships. The main focus is on processing and analyzing large-scale biological data. This involves developing algorithms and software to interpret and organize the vast amounts of information generated by modern biological experiments. Before diving into complex algorithms, let's cover the fundamentals of DNA sequences. DNA is composed of 4 nucleotide bases: adenine, represented by the letter A; thymine, represented by the letter T; guanine, represented by the letter G; and cytosine, represented by the letter C. These bases form the building blocks of DNA. An example sequence could be ATCG, which is just a string of these letters representing the order of bases in a DNA molecule. Now, let's explore some sequence alignment algorithms. First, the Needleman-Wunsch algorithm is a global alignment algorithm using dynamic programming, which seeks to find the best alignment across the entire length of 2 sequences. It has a time complexity of order mn and a space complexity of order mn, where m and n are the lengths of the sequences. Second, the Smith-Waterman algorithm is a local alignment algorithm for finding similar regions within sequences, regardless of the overall similarity. It also has a time complexity of order mn and a space complexity of order mn. Let's explore the dynamic programming approach with a snippet of code. Here's a basic structure for aligning sequences. The align sequences function takes 2 sequences, sequence 1 and sequence 2, as input. It initializes a dynamic programming matrix denoted as dp, with dimensions based on the lengths of the sequences. The base cases are initialized by assigning gap penalties to the first row and first column of the dp matrix. Each cell I, j represents the optimal alignment score between the first I characters of sequence 1 and the first j characters of sequence 2. This approach allows us to efficiently compute the best possible alignment between 2 sequences by breaking down the problem into smaller, overlapping subproblems. Let's learn about the BLAST algorithm, which stands for Basic Local Alignment Search Tool. It is a heuristic algorithm for finding regions of similarity between biological sequences. Key features of BLAST include fast database searching, statistical significance scoring, and support for multiple sequence types. This tool is widely used in bioinformatics for identifying homologous sequences and understanding evolutionary relationships. Phylogenetic analysis is the study of evolutionary relationships between species using DNA sequence data to construct phylogenetic trees. These trees visually represent the inferred relationships. Common construction methods include maximum likelihood, which estimates the most likely tree given the data, neighbor joining, a fast and efficient method for building trees, and maximum parsimony, which seeks the simplest tree with the fewest evolutionary changes. Gene prediction algorithms are computational methods used to identify genes within a DNA sequence. Common computational approaches include ab initio methods, which predict genes based on sequence features alone, comparative genomics, which uses similarities between different genomes to identify genes, and evidence-based prediction, which integrates experimental data such as express sequence tags to improve accuracy. Key features of these algorithms are the ability to identify coding sequences, splice sites, and regulatory elements in genomic DNA. Many gene prediction tools use hidden Markov models and machine learning techniques. There are many tools that come under bioinformatics. Some of the popular tools are NCBI BLAST, which is used for sequence similarity searching. Then comes ClusTAL, which is used for multiple sequence alignment. Finally, MEGA, which is for phylogenetic analysis. There are many programming languages in bioinformatics like Python using BioPython, R using Bioconductor, and Perl using BioPerl. Computational biology has numerous current applications including personalized medicine, drug discovery, disease diagnosis, and evolutionary studies. In the future, we can expect to see even greater advances through the integration of artificial intelligence and machine learning, real-time sequencing technologies, multi-omics integration, and cloud computing. Computational biology continues to revolutionize our understanding of life through advanced algorithms and data analysis. If you like this video, hit that like button and don't forget to subscribe. Visit CodeLucky.com for more such useful content.